Think of your body as a high-stakes construction project. To build something perfect, you don't just add materials, you have to tear down the mistakes. This is the story of apoptosis, the precise, programmed suicide of a cell. Every single day, billions of your cells die. It sounds alarming, but it's actually essential. If your cells didn't know how to die, you wouldn't have separate fingers, you'd have webs of skin. More importantly, cells that refuse to die when they are broken are the very cells that become cancer. Apoptosis is often called programmed cell death. Contrast this with necrosis, which is accidental death from injury. While necrosis is messy and causes inflammation, apoptosis is a neat, orderly, and energy-dependent process that dismantles the cell from the inside out without bothering its neighbors. To qualify as apoptosis, a few things must be true. It's a physiological process, meaning it's supposed to happen. It doesn't cause inflammation because the cell's contents never leak out. It requires ATP, it's an active process, and it's all governed by tight genetic control. There are two main ways for a cell to receive the death order. The intrinsic pathway is triggered by internal stress, like DNA damage. The extrinsic pathway is triggered by external signals landing on the cell's surface. Think of it as suicide versus execution. The intrinsic pathway is also known as the mitochondrial pathway. It starts when the cell realizes things have gone horribly wrong on the inside. The most common trigger? Irreparable DNA damage from radiation, toxins, or a lack of oxygen. Meet P53, the guardian of the genome. When DNA is damaged, P53 levels rise. If the damage can't be fixed, P53 stops being a repairman and starts being a judge, sending the signal that it's time for the cell to go. P53 activates the BCL2 family proteins. This is a constant tug of war. Pro-apoptotic proteins like BAX and BAK want to open the door to death, while anti-apoptotic proteins like BCL2 try to keep it shut. If BAX and BAK win, they punch holes in the mitochondria. Once those holes are punched, we reach MOMP, mitochondrial outer membrane permeabilization. This allows cytochrome C to leak out into the cytosol. It's like a chemical spill that signals the beginning of the end. In the cytosol, C-cytochrome C teams up with APAF-F1 and DATP to form a massive wheel-like structure called the apoptoson. This complex is the assembly line that activates our first initiator caspase. That initiator is caspase 9. Once the apoptoson wakes it up, caspase 9 is ready to start the chain reaction that will eventually dismantle the entire cell. Now let's look at the other route, the extrinsic pathway, or the death receptor pathway. Here, the cell might be perfectly healthy, but the body decides it's no longer needed, like a T cell that's finished its job. This pathway relies on death receptors, like FAS or the TNF receptor. A specific signal, the FAS ligand, binds to the receptor on the cell's surface, effectively handing the cell its walking papers. Once the ligand binds, the receptors cluster together and recruit adapter proteins. Together, they form the DISC, the death-inducing signaling complex. This complex directly activates our second initiator. For the extrinsic pathway, that initiator is caspase 8. It's the spark that sets off the same final fire as the intrinsic pathway. To keep these initiators straight, remember this: 9 is mine, because it's the internal mitochondrial pathway, and 8 is at the door, because caspase 8 is activated at the cell's outer membrane. Whether it's caspase 8 or caspase 9, both pathways eventually meet at the same point. They both activate the executioner caspases. This is the point of no return. The executioners are caspases 3, 6, and 7. Caspase 3 is the heavy lifter, shredding DNA and structural proteins. Caspase 6 collapses the nucleus. They are the demolition crew that ensures nothing useful survives. The cell begins to shrink. Its membrane starts blebbing or bubbling, and the DNA is chopped into fragments. Finally, the cell breaks apart into small, membrane-wrapped packages called apoptotic bodies. Here's the genius of the system. Those apoptotic bodies send out "eat me" signals to nearby scavenger cells. They are swallowed whole and digested. No leaks, no mess, and zero inflammation. It's the ultimate cleanup. When apoptosis goes wrong, the consequences are severe. Too little apoptosis allows damaged cells to survive and multiply, leading to cancer. Too much apoptosis and you lose healthy cells you need, which is what happens in neurodegenerative diseases like Alzheimer's. So remember, apoptosis is life's way of maintaining balance. It requires the precise coordination of sensors like p53, gatekeepers like the BCL2 family, and molecular assassins called caspases to keep you healthy. Understanding these pathways isn't just about biology. It's about understanding the delicate dance between life and death that happens inside you every second of every day.